Mole rat.
Yeah, longevity.
Oh,
Longevity.
I was going to say, okay,
Okay,
gotcha.
we're live. Hello, everyone. Welcome back. Thanks for joining us. We do appreciate you being here.
We do, yes. And one of us is in Michigan and the other one is in California. So here we go.
All right.
And
We're
you're Steve
excited.
and I'm Scott.
Right, and together we're back here, our musings, and here we go. Science and technology.
Yep.
Scientists have long studied unusually long-lived animals for clues to aging, but applying those insights has been difficult. Now, researchers report that inserting a single naked mole rat gene into mice extended lifespan and improved health. Researchers at the University of Rochester have taken a bold step in exporting longevity biology from one mammal to another. By moving a longevity gene from naked mole rats to mice, they improved the animal's overall health and modestly extended their lifespan. It probably, with snake oil, would give them more muscles too. An experimental result that hints at how nature's most resilient species may reveal new levers for healthier aging. Would you like to know more?
Okay, I'm a simple guy here. I did not know mole rats wore clothing.
So, they're naked, so they don't wear clothing.
Okay, does it have to do with their fur or lack of that they are called naked? I don't know.
Yeah.
Okay, here we go. Quote, our study provides a proof of principle that unique longevity mechanisms that evolved in long-lived mammalian species can be exported to improve the lifespans of other mammals. This is according to Vera Gorbanova, the Doris Johns Cherry Professor of Biology and Medicine at Rochester.
In the team's 2023 Nature study published August 23rd, 2025, not sure why there's a 2-year disconnect there, the key genetic change boosted production of an unusually large form of hyaluronan, high molecular weight or high molecular mass. Hyaluronan acid, and there's the formula there that I'm not even going to say, mice carrying the naked mole rat version of the gene had an approximately 4.4% increase in median lifespan, but that's probably hours for the mouse, alongside multiple markers of healthier aging. Ooh.
Okay. Naked mole rats have become a centerpiece of aging research because they combine an exceptional lifespan with an unusual resistance to many age-linked diseases, including cancer. Over years of work, Gorbanova and Andrei Soluanov have traced part of that resilience to how do you pronounce it? Hyaluronate?
Ronan.
Okay.
Yeah.
A gel-like component of the extracellular matrix that helps shape how cells communicate, repair damage, and respond to stress.
Hyaluronan is not inherently good or bad. Its effects depend heavily on its molecular weight. High molecular weight forms are often associated with anti-inflammatory tissue protective behavior, while smaller fragments produced when hyaluronan is chopped up can behave like danger signals that amplify inflammation and, in some contexts, support processes involved in tumor progression.
Future therapy can't simply aim to increase hyaluronan. It's the same stuff that we're talking about, so multiple pronunciations. It would need to preserve the high molecular weight form and avoid shifting the balance towards smaller, potentially pro-inflammatory fragments.
So just so the folks know how difficult this is, I'm going to spell it out for them. Hyaluronan. Now you try and pronounce it at home. All right. To test whether this mechanism could be exported, the team introduced the naked mole rat version of hyaluronan synthase 2 into mice, and they're calling it HAS2. All mammals have HAS2, but the naked mole rat version appears tuned toward producing unusual, large, abundant hyaluronan.
Okay, the word of the day. The engineered mice were better protected against both spontaneous tumors and chemically induced skin cancer. They also showed signs of improved health with age, particularly reduced inflammation across tissues, a notable result because persistent low-grade inflammation or inflammaging is widely considered one of the central drivers of age-related decline.
The nature work also linked high molecular weight hyaluronan to age-related gut integrity. As animals age, the gut barrier can become leakier, allowing inflammatory triggers to seep into circulation. The engineered mice showed protection against this deterioration, consistent with the idea that the hyaluronan helps maintain tissue hemostasis rather than targeting a single organ in isolation.
I just can't even tell you what I'm picturing with all this leaky gut and yeah. Anyway, this may not be one species fluke. Follow-up comparative work has reported abundant high molecular mass hyaluronan across multiple subterranean animals, often absent in closely related above ground species. So now we're talking about below ground. Okay.
Yep. That pattern suggests hyaluronan may be part of a broader evolutionary toolkit for surviving long lives under harsh conditions, potentially shaped by changes in both hyaluronan synthesis genes, HAS2, and degradation pathways, including HYAL family genes.
Even the researchers emphasize that gene transfer is not the end goal. Quote, it took us 10 years from the discovery of hyaluronan in the naked mole rat to showing that hyaluronan improves health in mice. This is according to Gorbanova. Our next goal is to transfer this benefit to humans. Oh boy.
Man, this is getting really interesting. This is getting fascinating.
Okay.
The group has laid out two practical routes: increase the synthesis of hyaluronan or slow its breakdown. Quote, we already have identified molecules that slow down hyaluronan degradation and are testing them in preclinical trials, Seluniov says.
Okay, a concrete example of that slow the breakdown strategy appeared in a 2024 scientific report study co-authored by Selunov and Gorbanova. Using a high throughput screen for, oh my gosh, hyaluronase.
Yep.
Okay, inhibitors. Enzymes, this is what they are, enzymes that degrade hyaluronan. Okay. The researchers had identified delphinidin and anthocyanin pigment found in various fruits and vegetables, such as a promising candidate.
I told you this is getting fascinating. I told you.
Yeah.
In the study, delphinidin mediated hyaluronidase inhibition, say that 3 times fast, increased high molecular weight hyaluronic acid in cells and mouse tissues, reduced migration invasion in multiple cancer cell lines, and suppressed melanoma metastasis in mice.
Oh boy.
Wow.
That
Wow.
This is especially relevant. This is especially relevant because naked mole rats appear unusual not only for how much hyaluronan they make, but also for how slowly they break it down, meaning their biology may be weighing on both sides of the equation. Okay, go mole rats.
There you go. Boosting hyaluronan seems to tilt aging toward a healthier trajectory, less cancer, less chronic inflammation, more resilient tissues, yet it is unlikely to be a universal shield. In July 2025, Biorixiv, a preprint added an important nuance. Mice expressing NMR HAS2 showed improvements in several late life health measures, but did not show protection from age-related hearing loss under the conditions tested. I can just see him with putting little speakers over the mice's head and just blaring Metallica, hinting that some organs may be less reachable by this pathway than others.
The Rochester work supports an increasingly testable idea. Some longevity adaptations may be portable, but tuning them into human therapies will likely depend on precision control, maintaining the right molecular form of hyaluronan, targeting the right balance of synthesis versus degradation, and watching carefully for trade-offs as different tissues respond in different ways. This has got Big Pharma written all over it.
It does. It does. There you have it, folks. The naked mole rat and big pharma.
Yep. When you get the opportunity, donate your money to the hyaluronan study, whatever, and go Big Pharma. But yeah, hopefully this, I don't know. I hope the mice like the Metallica. So you're frozen. I guess we're done. Peace. Perfect timing.